The Cumming Global Centre for Pandemic Therapeutics would like to acknowledge the traditional custodians of the land on which we've recorded this podcast series, the Wurundjeri people of the Kulin Nations, and we pay our respects to all elders past, present and emerging. Off Script is brought to you by the Cumming Global Centre for Pandemic Therapeutics, part of the Peter Doherty Institute for Infection and Immunity. I'm Dr. Matt Agnew, your host. I'm a proud Melburnian, and honestly, it's pretty exciting to know that cutting-edge medical innovation is happening right here in my own backyard. Dr. Amanda Capels has been Victoria's lead scientist since 2016, and in that time, she's played a key role in growing Victoria's thriving medical technology sector. In May 2021, Amanda became chair of the Science Advisory Group in mRNA Victoria. mRNA is a platform technology, and that's where things get interesting. Very excited to have you joining me today, Amanda. Thank you for lending us your time this afternoon.
Thank you, Matt. Pleasure to be here.
If we're naming Melbourne's true icons, we've got to be naming the G, the MCG, the Yarra, and of course, world-class medical research. This is something that's been critically important to Melbourne and Victoria. Surely you'd agree with this.
I would totally agree with that. Yes, indeed.
Obviously, we've been at the coalface of that medical research around vaccinations during the pandemic. But do you want to share with us some of the really important medical research that's, has steeped Melbourne's scientific scene?
So Melbourne has a long-standing track record in health and medical research, and it started, in fact, well over 100 years ago, and we have travelled along with the development of the science, of the biology, that has enabled us to really have a very strong medical research community here in Melbourne, right across metropolitan Melbourne. And then from a state government point of view, as Victoria's lead scientist and formerly as the director of biotech, we've doubled down to ensure that our top quality science has access to the tools, the technologies and the translational capabilities that are needed to really transform the research into clinical trials and into products that can help patients.
And that's such a key and critical part, right? The translation from cutting-edge medical research to actionable, tangible, real-world impact.
Absolutely. So the steps that need to be taken from the lab into the clinic are multifactorial. And so what we have identified in collaboration with our researchers and with industry are the key pieces of technology where we can add value to accelerate the translation of the biology to products and services.
Fabulous. And we'll kind of dive in a little bit more to something that was more closely adjacent, I guess, to the pandemic. So you're also the chair of the Science Advisory Group at mRNA Victoria, the government agency tasked with building Victoria's mRNA industry. mRNA is a platform technology, a bit like IKEA furniture, with modular components that can be prepared in advance and quickly assembled to respond to different viruses. The technology was behind the rapid development of the Pfizer and Moderna vaccines that people would be most familiar with, the COVID-19 vaccines. Can you explain a little about how mRNA technology works and I guess how it differs to, what's being considered more traditional attenuated virus vaccinations?
mRNA works by providing the cells of the body with the instructions to make a protein. And in the case of mRNA vaccines for COVID, the protein that is made by the vaccines migrates, if you like, to the cell surface. And what it does there is it's identified by the immune system. And so the concept of the vaccine is really to prepare the body to better respond, for the immune system to respond and to negate, if you like, the effects of the virus. So It's not an antiviral, which is why we need other forms of technology for infectious diseases. But what it does is modulate the immune system so that we're better prepared. And so antivirals will, the idea or the concept behind antivirals is to interfere with the replication mechanism of the virus. So they directly action against the virus. So this is why, for those patients who had a compromised immune system, why antivirals were essentially were really, really important as adjuncts to vaccination. So some people with an intact immune system don't have a requirement for antivirals, but those that do it was an important step in helping them recover from the infection.
Right. And so mRNA, we're giving instructions to the cells and helping inform how the immune system battles these viruses.
So with antivirals, for example, it can take years actually to really identify a molecule that will have the efficacy and the safety that is required. But the precision with mRNA, shortens the time frame. So from a productivity point of view, it's huge.
Yeah. So obviously we'll be critically instrumental in future pandemics because of that rapid response and identification period.
That's right. It really gives us a very sharp tool in the toolbox for us to intervene against potential further pandemics.
The M in mRNA stands for messenger because the mRNA contains instructions for cells. So, and we kind of talked a little around it, but we'll go into the guts of things now. What instructions specifically did the Pfizer and Moderna vaccines have for the body?
So the vaccines for COVID really targeted the spike protein on the virus, and so that is the protein. So when you think of a virus, it's and many people have seen the diagrammatic representations of a little ball with spikes around it.
Yep.
And those spikes are actually proteins that attach to the host cell, and that allows the virus to penetrate the cells and causes the problems of infection.
It was that sort of binding and allowing the virus to access the cells that was inhibited by the mRNA vaccine. That was the instruction. So yeah, and I'd be interested to know your thoughts on this. The mRNA technology had been understood for a while. Did you see COVID as sort of it kind of expedited bringing it to a rollout publicly and actually seeing it in action? Is that something that you'd sort of agree it may not have been rolled out still if it wasn't for the pandemic sort of forcing the hand, so to speak?
I think, absolutely the pandemic brought forward the development of the technology. So as I said earlier, it had been investigated for the treatment of cancer for quite a number of years. And but nevertheless, it really brought focus to the area and catalysed a whole range of activity both here in Melbourne, but across the world for a range of infectious diseases, as well as looking at other applications of the technology. So it was sufficiently ready at the time of the pandemic to be brought forward in the way that it has by both Moderna and BioNTech. And the success of and the demonstration of the technology and its usefulness has really stimulated a wide range of other activities. And that's important because there are other infectious diseases of concern that may not have such a big market as we experience with COVID. And I'm thinking of infectious diseases in the Asia-Pacific region, in Africa, where the market size is not sufficient for major pharmaceutical companies to develop new products. But with the precision, the speed, and the utility of mRNA, for the first time, it allows us to approach those conditions and really have a huge impact on public health around the world, but particularly for low and middle income communities.
In terms of our mRNA technology outside of vaccines, its applications as a therapeutic, is that something that is being looked into, and is it something that's being funded here in Victoria by mRNA Victoria?
So in terms of therapeutics, so mRNA has the potential to, and it is being developed as a therapeutic. And in fact, if you think of it in terms of the COVID vaccine, it doesn't prevent infection, but helps the body deal with it. And so mRNA is an immunomodulator in that sense, and that's how it's used in the treatment of cancer. So there are a range of indications, if you like, where the potential for it to act as an immunomodulator, as a therapeutic, as an immunomodulator is relevant. We typically think of therapeutics as those that intersect more directly, and apropos the earlier conversation around antivirals, and so in the case of mRNA, where it can really have promise as a therapeutic in that sense is in those conditions where there is an absence of protein, because when you think about it, how mRNA works is by providing instructions for the cell to make a protein. And there are a number of conditions where there is either an absence of protein or a low level of protein. And so being able to make more of that protein is useful for that indication. But of course, that then requires, there's an additional set of complexities there, is that mRNA traditionally is short-lasting, so it doesn't persist. And so to be able to treat to use mRNA as a therapeutic, you need to consider how you can produce the protein at sufficient levels, so both the quantity of the protein, how you can provide that at the site of action, and how you can provide it for long enough to have the desired effect. So you can see already it's quite a number of considerations that go into mRNA as a therapeutic.
So in terms of mRNA Victoria, what are the key areas where a lot of resources and money is being focused at this moment?
So mRNA Victoria was established 4 years ago when it became apparent of the potential of mRNA. And so we were able to build on our previous work in biotech. And what we did was to establish a dedicated team within government to really catalyse and accelerate the development of our mRNA ecosystem. So what we did was to invest in a pipeline of mRNA candidates, so both for COVID, but for other infectious diseases and for cancer. So it was important for us to be able to accelerate those new discoveries. Alongside that, we invested in the associated infrastructure that you need that is not candidate specific but related to the platform technology. So looking at the immune response to mRNA is an important consideration in developing a potential candidate. And then thirdly, it was in terms of manufacture. So having a pipeline of mRNA candidates is of limited value if you don't have the translational capability to make the material for testing in animal studies and in clinical trials. And so mRNA Victoria secured the Moderna facility, which provides 100 million dose vaccine capability down at Monash University. And then more recently with BioNTech, we have, if you like, a foundry of mRNA capability with a production facility being built currently out at La Trobe.
I was going to just follow up. Is that something that we're looking into, having our own manufacturing capability? And it sounds like absolutely.
Absolutely. So we've covered the field. We have manufacturing for a clinical application, so for population use, and we have a facility that will accelerate the translation of discoveries into candidates that can be tested.
Fantastic. So we've got the whole life cycle from research, development, clinical trials, all the way through to manufacturing and rollout, which I imagine will be tremendously important, not if, but when the next pandemic rolls in. Thank you so much, Amanda. This has been a fantastic chat. And I was completely unaware about the breadth of application of mRNA technology. So thank you so much for your time.
Thank you, Matt.
As we've just heard, mRNA, a platform technology, was critical in the development of COVID-19 vaccines. But it's not just vaccines that this impressive technology can be used for. It can also be used to develop pandemic therapeutics. And a researcher at the forefront of that is Dr. Paula Saval, who's supported by the Cumming Global Centre for Pandemic Therapeutics. Paula, lovely to have you here. I'm going to jump straight into this, and I really love this analogy that you've used. You've likened mRNA technology to a mail delivery driver delivering a message to our cells. Can you explain that to me and our listeners, of course?
Yeah, it's been an analogy that's worked well. It sounds a bit crazy, but when you get into it, it really does make sense, I think. So the mRNA really is like a recipe book. It has a message. It encodes a message, or it contains a message that our cells need to receive in order to then produce something that could be therapeutic and could have a therapeutic effect. So however powerful this RNA molecule is, it's very fragile. And in and of itself, it doesn't know where to go. And it can't actually get into the cell, which is where it needs to be in order for the message to be read. So it's essential that this mRNA gets into the cell and that it gets into the right cell as well. And that is where we need this mailman delivery. So essentially, we need to make a capsule or a little box around the mRNA that does a couple of things. It protects it. It protects this very fragile mRNA, but it also delivers it to the right place. So this capsule, which in complicated terminology is called a lipid nanoparticle, is the mailman, if you will.
And just quickly for listeners, a lipid nanoparticle is fat? Is that kind of?
Yes, it's a little fat bubble.
Yep.
Made up out of fats that we mix together, and these fats sort of surround. Looks like a soccer ball with the mRNA inside of it. So it's usually spherical, but it can take different shapes depending on how scientists design it. And this soccer ball containing the mRNA, all of this is like at a microscopic scale, right? It's something that we can't see with the naked eye. We can't even see it with a microscope. It's so small, but we can make it in a lab, which I find magic. And we need that soccer ball to deliver the mRNA to the right cell.
I mean, it definitely sounds like magic, which a lot of, I guess, impressive and complex science, often it's indiscernible from magic, right? Let's just go into an even more fundamental question. What is RNA?
RNA is part of what we call the central dogma of biology. So the world wouldn't be without RNA. We wouldn't live without RNA. And the role RNA plays in biology is often that of a messenger. So we have DNA in our cells that contain our genes. And these genes contain all the instructions of how to build life and everything in life. But in order for that gene or the book to be read, it needs a messenger to take those instructions to other parts of your cell, of your body, that can then action those instructions. It's like a recipe book that needs to be delivered to the chef so that the chef can go and make us a very nice meal. And so the mRNA is that in-between step, which is why it's often called a messenger RNA or mRNA.
Right. So the messenger is essentially informing the RNA what to do. It's handing over the instructions to the mRNA. Yes. And so has mRNA kind of been your research for a lot of your research career, a lot of your academic career? Has this been your wheelhouse, the mRNA field of research?
It has become my area of research. It wasn't initially. I think we've known about mRNA for a long time. We've known its role in biology generally. But I think only more recently we've become more aware of the therapeutic potential of mRNA. So previously, when we are trying to design new therapeutics for any disease, we tend to work with different types of molecules along the lines of what you would have in your paracetamol. These are chemical structures as well, but from a very different entity than an mRNA molecule. And so only recently have we started to explore, can we use this messenger idea that already exists in biology, that is such a fundamental part of life? Can we utilize this? And what if we make a recipe? What if we make our own mRNA? And rather than have it be the instruction to just go about and do normal life, what if we build the instruction to make it into a therapeutic? And that idea has gained a lot more traction in recent years and is becoming adopted by more and more scientists.
Fantastic. We'll get into the usage of mRNA as a therapeutic in a moment, but I might just go back in time a little bit. Obviously, the first exposure, I guess, most of the general public had to mRNA was rather than in a therapeutic capacity, but in a vaccination capacity. And for you, were you involved in mRNA research at the point during COVID when it started, taking that idea, that research, cutting-edge, state-of-the-art research into a mass-produced product that was being disseminated through to the general public for vaccination? Was this something that you were part of, or not that specific research, but within the mRNA field? And was it a really exciting time to see it come from research to fruition?
Yeah, absolutely. We were working on this. We were working on mRNA therapeutics already before the COVID-19 pandemic hit. The idea of using mRNA as a therapeutic stems from way before the COVID-19 pandemic as well. It's been subject of 2 Nobel Prize awards as well, or 2 people won the Nobel Prize on mRNA technology. So this idea stems from before the COVID-19 pandemic. But what the COVID-19 pandemic and the application there of mRNA has really helped us with is see it work in real life. So we've known this idea, we've hypothesized, we've had conviction that it would work, but to really see it happen and to see it also go through all the required safety steps and see that it's actually safe for use in humans in the grand scheme of things and can help us combat a pandemic like that has definitely made the world of difference and has really given us a lot more motivation to keep pursuing this kind of area of research.
Yeah, very, very exciting. I think that transition from research to, as you said, real world application, I think for a lot of scientists is really exciting because often we're fumbling around in the dark with things that stay in the world of hypothesis and research proof of concept for a long time. So I can imagine it was quite a profound moment seeing that translation happen. You're working on using the mRNA platform to develop a cure for HIV and also for respiratory pathogens, which I have some slides which I read just before and got very excited. So we will get into that a little later. But maybe we'll start with the development for the cure for HIV and using mRNA. Can you talk us through that?
Yeah, so I think this all sounds like we're trying to use this one and the same mRNA molecule for this range of therapeutic applications, right? And it sounds sort of difficult to comprehend. How can you use one and the same thing for all these different applications? And so it's important, I think, to go back to that analogy of the mRNA being a recipe book that can provide instructions for how to cook a beautiful tiramisu for dessert, but it can also be a recipe for how to make a cottage pie. And for both, you need a recipe. So the mRNA is the same, but we scientists can redesign the mRNA to be recipe for different things, right? And so we can utilize this technology for whatever we want, even though they're completely different pathogens. They infect us in different areas of our body. They affect different cell types, etc. So you can have a wide range of applications, all using the same mRNA technology.
That's one of the quite strong upsides to mRNA tech, right? Is the ability to quite rapidly rewrite the recipe.
Yeah, I think that's a fundamental advance in science that happened at some point, that rather than realizing this mRNA exists within our cells, that at some point we figured out how to build this mRNA ourselves. And so for me, I don't do that myself. Thankfully, there's smart people out there who know how to do that. And the magic is that all I have to do is go online and type in my letter combination, which goes like CTG, AA, CC, etc. And then someone else builds this mRNA for me, and it gets shipped to me, and I can then use it. So I can order an mRNA that I think would be the right recipe to try and cure HIV. And tomorrow I can order an mRNA that I think will help us combat the next pandemic.
Right. And so it's as simple as there's sort of a supply chain and ordering mechanism where you can go online and just place an order, basically.
It actually is. Yeah, it's magic. And apparently now it's very easy to build these mRNAs. So that process is relatively quick. And that is also in part why we were able, I wasn't involved, but why science was able to develop these vaccines during the COVID-19 pandemic so rapidly. Because once we realized what the virus looked like that was causing the pandemic, we can then design the appropriate recipe, the appropriate mRNA, and just go online, order that. And that was the first pilot vaccine that we could then go and test. That process and how rapid that is a massive advantage of mRNA technology.
Did you find during the pandemic and sort of some of the public response, because I guess for a lot of the general public, their thoughts about vaccine development is a decades-long process, this incredible new technology, I mean, in this case, it was a couple of 100 days, or it was less than a year, I think, from the event to developing the mRNA vaccine. Was that something you found challenging at times?
I mean, I wasn't involved in it myself in that sense, but I've definitely become aware of some of these sentiments within the general public. And I think it's completely understandable because I think the COVID-19 pandemic was one of the first examples of where we used something like this, which we tend to call platform technologies. So it's essentially a therapeutic that's made up out of different Lego blocks that we put together. And so if the individual Lego blocks have been validated already, the combination of that and the final therapeutic can be processed through all the required regulatory and safety steps much more quickly because we already know its parts.
Right.
And I understand that that has been something that maybe us scientists needed to communicate better to the general public. And I think we're going to see more of these vaccines or more of these therapeutics go through those regulatory processes and be tested in the clinic. And I hope that at least over time we can explain this a bit better. And I feel that we scientists owe that to the general public.
That was a good segue into mRNA and its applications in a therapeutic capacity rather than a vaccination capacity. I have some slides from a talk I believe you gave very recently, which I got quite excited about. I was aware of, using these lipid, what was the scientific term?
Lipid nanoparticles.
Lipid nanoparticles for enclosing the mRNA in this sort of fat ball that then your cells could accept, I guess, to receive the instructions. This is talking about, in a therapeutic way, maybe we'll go by, what's one of the big challenges with therapeutics? It's in terms of administering the therapeutic. Would that be correct?
Yeah, I think that's absolutely correct. It's also one of the biggest sort of steps we have to make to change this technology from being a vaccine to this technology being suitable as a therapeutic. Think about it. We've all received these mRNA lipid nanoparticles as an injection into our arm. But for most therapeutics to work, they may need to go to different areas of your body. So we might need to inject this as a therapeutic into the bloodstream. Some therapeutics are taken orally. In the case of the next pandemic, the virus tends to be what we call respiratory, which means it affects our airways. And so it's probably going to be very beneficial if we can deliver this mRNA recipe to different areas of your respiratory system. So your upper airways, your nose, and your mouth cavity. and then also in your lungs. And delivering it there is going to be a challenge, And also the main difference from what we know already, right? We can't just inject it into your arm and hope it's going to end up in your lungs. That's not how it works, unfortunately.
Yeah, so and this is essentially taking that concept of the tiny fat bubbles injected in that is absorbed by your cell to creating a sort of like a vapor mist.
Yes.
Would that be, and the mRNA is in these kind of tiny airborne particles that you then inhale.
Yes.
And then that's sort of directly administered to your lungs.
It's kind of what has made this pandemic or the past pandemic so tricky is that the virus transmits via little aerosols. And therefore, if I cough and I'm in the same area as you are, that you might become infected. We're trying to actually utilize that fact, right? And so rather than having, because the reason why this works for the virus is because we inhale these aerosols and they go everywhere into our lungs. Well, what if we can make aerosols that contain something good? And so the aerosol travels, you inhale it. Naturally, that means that the therapeutic is deposited everywhere throughout your lung, everywhere where it needs to be.
I love that. The kind of, this is the, I guess, vector of attack of the virus. Let's repurpose that.
Yeah.
Administering the therapeutic. So I guess, from that idea, what are the critical next steps to translate that research into a patient-ready therapeutic, I guess, ideally before the next pandemic arrives?
Yeah, so there's a lot of things, to be very honest, that we need to do. This is why our research is part of a broader center, the Cumming Center, and a lot of people are studying different aspects of this technology. So one area of research is to redesign this fat bubble and try and see if there's different fats we can use. Can we mix them in a slightly different way? There's also areas of study within the center that look at not using a fat, but using other chemicals that we can maybe mix into this soccer ball and build better cages that are not just a lipid nanoparticle, but other types of nanoparticles. So that sort of chemical design is an important aspect, and how can we optimize that? That's one. The second, we've already looked into how to deliver it to the right place. And one thing we really want to do there is try and come up with different designs where, for example, one goes to the deep lung, whereas a different design may stay in your nasal cavity. And so if we just have like an arsenal of different delivery methods ready to go, when the next pandemic hits, we can just pick the right one off the shelf and go with that. And the third area, I think, is around that sort of delivery spray and the aerosolization, where we'd really like this to be something that people can use in the comfort of their own homes, and they don't need to go into a hospital and use a very complicated device to then sort of be able to inhale that therapeutic, right? So we need to reformulate a therapeutic into something that could be put into a nasal spray bottle or into something of a mask that you can use at home to have the therapeutic aerosolized and for you to then inhale it. And so that is also a big aspect of work.
Right. I like the arsenal analogy where I guess you're kind of creating a menu to select from what type of, using that ordering thing that you said to create the mRNA, what the structure of the lipid fat ball, or whether it's something more exotic, the administration method, and you can basically bespoke create whatever is needed in a therapeutic way to address. And I guess that's because you're unable to predict what the next pandemic's going to be. You're kind of at the mercy of it could be anything. Is that why this arsenal is so important?
Yeah, I think it is. And I think, look, we have a lot of ideas about what it could look like. The main area is a respiratory pathogen, which means something that, again, transmits through our airways. But that still leaves so many options. And I think you're always better off if you're just prepared and have a wide range of possibilities ready to go. Even if you think you may only need the first 3, I think you're always better to have 10 backups than to pick just those 3 and only prepare for those and then be caught off guard.
Got one more question here. And I'm excited to hear your answer for this one, actually. What makes platform technology so exciting in the development of pandemic therapeutics?
I think for me, platforms and platform technologies equal speed. And I think speed is of absolute essence if we want to prepare for the next pandemic. Because we're preparing for something that is currently yet unknown, we don't really know yet what we are preparing for, yet we need to become prepared, right? Somehow. How do you do that? So the more of these building blocks that we can already develop now, and the more of those we can already validate, and with that I mean we know that it works and we know that it's safe, or up to what extent is it safe, or what are the potential side effects. The more we know already, then once the pandemic hits, it's a matter of putting puzzle pieces together. But they are now known puzzle pieces that we're putting together. Whereas otherwise, the pandemic would hit and you would have to start from ground zero, which I just don't think is a smart idea. So I'm a huge fan of platform technologies. And I think the mRNA in and of itself is a platform that is super rapidly adaptable. This mailman that we've built around it, the lipids or the other chemicals that you put in there are a platform in and of itself. But even the instruments that you could use to deliver these, like the nasal spray or the mask or whatever it is also a platform. And we can develop all of these separately and simultaneously. And then when required, you add all of them together and there you go.
Right. And it's like you mentioned earlier with the Lego blocks, where if you know all these Lego blocks are safe, there's a good chance together
exactly
they'll be safe.
And it doesn't mean that we don't need to then still test that combination of the Lego blocks, because we do and we will, but you can already sort of accelerate the process because you've got some fundamental understanding of the individual Lego pieces, which I think will make the world of difference.
Fantastic. And is there anything else that you'd like to add that we haven't touched on?
Good question.
Good question.
Very good question. I think one other thing that is good to mention with regards to the platform technologies is that the Cumming Global Center is really unique in how it's set up, in that it's given a lot of attention to these different platforms. And being part of this center and being united in such a clear mission, in that everyone that's part of the center is eventually working towards the same mission, it really helps foster this sort of collaboration. And look, if you just focus on that platform, I'll focus on this platform, and we keep each other updated. And we know that in however many years' time, we can come together and put our puzzle pieces together. I think that's a unique way that this funding and this center has been designed.
Fantastic. Well, I think that covers everything to chat about today. Thank you so much for joining me, Paula. This has been a fabulous chat.
Thank you. It's been a pleasure to be here.
As we've just heard, when we talk about pandemic therapeutics, there's a whole journey from early platform technologies like mRNA all the way through to getting a treatment into patients' hands. That includes everything from figuring out how the drug should be delivered, to running clinical trials, to navigating regulation and scaling up manufacturing. It's a huge, complex pipeline, and it needs serious investment and coordination to work in a crisis. One organisation doing exactly that is New York's Global Health Investment Corporation. They're a non-profit dedicated to accelerating the development of technologies aimed at improving global public health. And Devon Sock is their head of science. Hello.
How's it going?
Good. How are you, Devon?
Doing all right. How you doing?
I'm good. I'm Matt. All righty. I am joined right now by Devon Sock, who is the head of science at the New York's Global Health Investment Corporation. Have I got that correct?
That is correct.
That's correct. But you're not in New York. You're in San Diego.
I am in sunny San Diego.
I like it. Keeping things interesting. Wonderful. Let's dive straight into it, Devon. Why is investing in platform technologies like mRNA so critical to making sure that we're ready for the next pandemic?
Yeah, I think it comes down to, I think the simple answer is we don't know what the next pandemic is going to look like. And either that pandemic is either going to be caused naturally, it could also be accidental, or it could also be intentional. So I think just to contextualize it a little bit, I think if we look through the history of pandemics, starting at the early days of, the 6th century, 14th century bubonic plague, that was a bacterial outbreak. Following that, there was the flu. So Spanish flu, 1918 flu, that also killed millions of people, up to 100 million people. That's an RNA virus. And then following that, we had HIV, AIDS, which has now killed up to 40 million people. That was since 1981. That was a retrovirus. And then, of course, COVID-19 in 2020, which we're all familiar with. And in addition to that, those are the big pandemics. There is also a bunch of epidemics that have happened that were fortunately contained for a number of different reasons, didn't become global outbreaks. That include Ebola in West Africa, SARS, the original SARS, MERS, Zika, Nipah, Lassa fever in West Africa, and measles. So I think all that to say is there's a lot of bugs out there. There's a lot of different pathogens. They all look different. They all have different profiles. And so it's really hard to predict what's going to come next. And so in the backdrop of uncertainty of like what we need to prepare for, the importance of investing in things like mRNA platforms is we need additional tools. So the more tools that we have, the more that we are able to address any potential outbreak that's going to happen. And so if we're, for example, limited to kind of conventional vaccine platforms, there's different varying efficacy of kind of existing vaccine platforms. There's different timelines, there's different safety profiles. And so having additional platforms like mRNA really adds to how we can effectively respond. And I think the incredible thing about RNA is it has two benefits. One is it's really fast. So in the scenario of a pandemic where really the timeline is really important in terms of containment and preventing future outbreaks, mRNA is the fastest way to produce a vaccine. And the additional benefit is that it's also really adaptable. So you can really plug and play different things that you need to really produce a vaccine for whatever the target is. So there's a lot of benefit to mRNA, and I think there's a lot of interest in finding kind of other adaptable platforms so that we can respond to whatever the potential threat is going to be that hopefully we can contain more effectively.
Can you walk me through how investment actually happens in terms of what does it look like to identify promising new technology like mRNA, and then how do you help move that towards real-world impact, which I guess the COVID-19 catalyzed that a little bit with regards to mRNA technology. So maybe with respect to that, but also more broadly.
Yeah, so I think there's there's a whole infrastructure and ecosystem. And I think the main takeaway or main message is all of it's important, and you need all of it to really bring something from discovery through to actual impacting people's lives. So starting kind of at the beginning, the majority of discovery research is born out of universities. And so all those universities are funded through typically public funding, government funding, and that helps to, and in the grand scheme of product development, it's probably the cheapest part. But there is a lot of, it's also the highest risk part. So there's a lot of things that fail. I was a discovery scientist, got used to a lot of failure constantly. It's part of the
scientific process,
the profile of a scientist who's willing to deal with failures.
Yep.
That's just discovery research. And so because of that, you actually need some longevity, you need some stability, you need institutions that are able to support scientists in the long term so that they can try out a bunch of things. And, 1 out of 100 things might actually hit, but you need to actually do that and have a space where you could be innovative and also have the majority of the training and work that's done in these research institutes are done by students. So graduate students, postdocs who are paid modest stipends to explore kind of research. And so there's a whole infrastructure there that generates discovery. And what happens is typically if there's a new invention or a new discovery, and this is to say that there's a lot of other discovery that happens in biotech and also in pharma and other kind of modalities, but just kind of speaking the more general terms. So coming out of like university settings, you then have an invention, and either that invention goes out and you license it so you give it to a product developer at a cost, and then you move on with your research. And the product developer moves that forward, or you decide that you want to spin out a company. And so you license that out to and create a startup. And so in that startup, you're now at the seed stage. And that's where you typically rely on either institutions that have funding to support seed stage companies, or you rely on angel investors, the people who've, were really successful in selling previous companies and really want to fund at the early stage of research. There's also some public funding grants to help support kind of that movement into the private sector in terms of funding startups. And during the seed stage is when you generate data. You generate additional data to try and attract kind of venture capital funding, which is more funding to kind of move things along. And so there's a lot of things that fail at that stage in terms of the data doesn't look very good, or you don't attract enough data to attract funding to kind of move it forward. But if you're lucky and the science works out and the timing works out and you meet the right VCs, venture capitalists, venture capital firms, then you start to raise for a Series A. And there's a series of different funding cycles from series, through the alphabet, A through F, where you raise buckets of funding to kind of progress your product through development. And so typically, once you have discovered something, you want to test it in vitro, in a petri dish, then you want to test it in animal models, and then eventually you want to test it in people. And the kind of people testing falls in 3 stages. Phase 1 is when you test it in safety. Phase 2 is when you test a little bit of efficacy, try to find the right dose, what's the right amount that you need. And then you have phase 3 trials where you test whether or not it actually works in terms of clinical endpoints and whether or not you have the outcomes that you're working for. The costs as you progress through that increases quite dramatically. And so the amount of funding that you need for each of those series and each of those stages increases quite dramatically. But if you are one of the rare few that makes it through all those stages and actually have a product that you prove is efficacious, then typically you have kind of 2 routes. One is you IPO and your company moves it forward and you make it available to people, you license it, regulators accept it, and you start to market it and sell it, which is great. And that's kind of the story of Moderna and the mRNA vaccine. Or you get acquired from large pharma. So that's mergers and acquisitions. And then the pharma takes on responsibility of distributing it. Once that you reach that stage, then you actually have to get it to people. You have to get people to what you want to take it, and then you have to figure out how it gets paid for. Reimbursements, insurance, kind of procurement packages. So there's a lot of things that fall into place from like when you discover something in a lab to really get to a point where you actually have something to give to people and actually have people use it to have impact. And each one of those stages could absolutely fail and often fail. And the process is actually quite rigorous because not a lot of things actually get through.
Yeah, I think that's it. And that rigor, I think, is really important to be communicated to the public to ensure and assure them that the vaccines are safe. They've not come out of nowhere. They haven't come up from a black box. They've gone through this really, really rigorous testing and trial process.
No, I think the future, despite all the uncertainty that we're operating in now, I think there's a lot of optimism in terms of the tools and technologies that we have that really can change human health. And I think the AI revolution and kind of the tools that are available to accelerate drug discovery, to understand biology, to understand human health, I think probably the big exciting thing that I think about in the next 5-10 years is this idea and concept of once we actually have AI and kind of data-driven approaches, traditional product development is really focused on the majority, right? Like you want to hedge your risk. So you produce products that can hit the most people. And there's a lot of people who are on the fringes who are different profiles who might not directly benefit from that. And there's a lot of reasons that go into that. The clinical trials are complicated, the endpoints are complicated, human beings are complicated, et cetera. But I think we're going to reach a point where you can really be selective in enrolling the right people into trials, be really selective in the design of interventions that are specific for groups of people, and be really specific in terms of product development. And I think once you get to that point, that what's interesting and what's exciting in the next 5-10 years is really truly realizing personalized medicine, where you don't have to always buck or trend towards the majority, but really develop things specifically for groups of people that are really going to be helpful for those groups of people. And I think it's both the economics and the technologies are going to allow us to actually do that. And I think that's where we're going to see a renaissance in human health.
One of the things we're really focusing on in our discussions is around therapeutics and the impact of therapeutics. So would love to hear your thoughts in terms of the impact therapeutics can make, certainly within the pandemic space, but broadly as well.
We talked about kind of the toolbox, of different platforms that are available to respond to different pandemics. We could think about vaccines and diagnostics and therapeutics as also a toolbox. I think therapeutics are a really important part of the toolbox, going back kind of to the history of pandemics, right? So bubonic plague is a bacteria. We don't really have a strong, efficacious vaccine for that yet, but we have antibiotics. And so therapeutics for HIV. I was working as part of a program on trying to discover and develop an HIV vaccine. It's been 40 years. We still don't have an HIV vaccine. It's probably going to be a while, but we have therapeutics to allow people to live. And so I think for each of these, again, going back to the diversity of potential different pathogens that we might be exposed to that might cause the next pandemic, we may have some tools in the toolbox to create a vaccine. We may not. And then in the case scenarios where we don't, having things like therapeutics available to be able to respond to those are going to be crucial. So I think it's a really, really key component, a core component of responding to any future threat or future pandemic.
Yeah, I think it's perfectly articulated when you said it's a toolbox. We've got a number of things within that toolbox. Vaccines are one of them, therapeutics certainly another. Ideally, all of them work for particular pandemics, so we can use several, but like you mentioned, HIV is the quintessential example where the vaccination tool isn't working, or certainly not yet, but the therapeutics are tremendously important. So thank you for your time, Devon. It's been wonderful chatting with you and hearing all about investment, and thank you so much for your time.
Of course, thank you so much.
As we've heard in this episode, medical technology like mRNA has the potential to transform pandemic therapeutics. That's great news for Australians, but it's also good news for people everywhere. In our next episode, we'll explore how pandemic therapeutics could improve lives across the globe, especially in countries where health systems aren't as well resourced as ours.